Hello, my name is Kenadie Janulewicz, and I have been working on advanced methods using nuclear magnetic spectroscopy with Dr. Pites. This here is our NMR machine. This is a sample of one of the tubes that we would use in our machine. If we open up our cover and spin the dial, our sample will pop out. We fit it with a sleeve and we can send it back down where it will spin in between some magnets. We'll close our cover, and then over here you can see our computer that is fitted with all of the software to run our experiments. Here's an example of what one of our experiments would look like, pulled up on our computer. Over here you can see a poster that compiles all of these, or some of the experiments completed for my honors project. Some of the first sets of experiments that we did were related to two-dimensional NMR. When looking at the goal of these spectra is to identify which carbon peak corresponds to each carbon on our structures. When looking at the hydrogen NMR, we are able to determine the difference between A, B, and C, but when looking at the carbon, we are unable to. The only one we can differentiate is H. The rest we will have to look at different runs to tell apart. The first one we will look at is carbon A in respect to B and C using our depth. It will show carbons with 3 hydrogens as up and 2 hydrogens down. So we know this carbon peak is A. To be able to tell the difference between B and C, we will look at our het core. We know from our hydrogen NMR that this set of peaks is carbon C, and so when we intersect our lines, we know that this peak will be carbon C, and therefore this one will be B. To be able to tell that this peak is E and that one is F, we use long-range coupling on our COSY. This coupling right here determines that this side is E. We are confident in the order of these carbons, even though we are not able to differentiate between D and G. The next set of experiments that we did are the calculations of percent alcohol in a variety of beverages. Each of these spectra will be the same, with a peak for CH3 and CH2 of ethanol, whereas the water and alcohol of ethanol will be an overlapping peak. To be able to determine the alcohol concentration, we will be able to use our CH3 or CH2 peak, as they are only from ethanol. They will be a third of the moles, or the alcohol will be a third of the moles or half of the moles, depending on which carbon peak we use. We came up with an Excel spreadsheet to help us with calculations, where we will use the areas of integration, which are highlighted in pink, to calculate our mass of alcohol. We arbitrarily assigned our alcohol and water peak to 1,000 to display more significant figures. Here we can see our CH2 peak came out to be 171 and 256 for the CH3. Using the molar mass and density of water and ethanol, we are able to calculate our percent by mass. We used a standard that was 35.2% by weight to check these calculations, and we came out with 32.4, which is close. However, since most alcoholic beverages are sold by percent volume, we went ahead and used our density to get 39.8% by volume. We applied these calculations to a variety of alcohols. Here are 3. For Glenlivet, we were able to calculate 40.2 with our CH2 peak and 40.4 with our CH3 peak, while the label read 40. For Chardonnay, we calculated 13.7 and 17.5, while the actual was 13.5. And then for vodka, we calculated 40.4 for both the CH2 and the CH3 peak, while the actual is 40. Another interesting set of experiments that we did is carbon coupling. So here we see a spectra of isopropanol where we ran it as coupled. So our carbon to hydrogen spectra is large of 138 and 121, and it takes up a large portion of the spectra. This is why we typically run carbons as decoupled like we did in our two-dimensional NMR. We also did this with trimethyl phosphate. Our carbon to hydrogen coupling is large at 148, and our carbon to phosphorus long coupling is only 6. This produces a quartet of doublets, but it's really interesting to see carbon coupled to something other than hydrogen. We also calculated the natural abundance of boron isotopes, and this is unique as boron does not have a nuclear spin of 1/2 like carbon, hydrogen, or phosphorus. Boron 11 will have a spin of 3/2, which will provide us with 4 peaks, whereas boron-10 will have a spin of 3, which will give us 7 smaller peaks. Using the areas of integration of these peaks, we were able to calculate the natural abundance, which we calculated 78.5 and 21.5%, and it is roughly 80-20 in nature, so we were really close. The last set of experiments we did are associated with water's effect on hydrogen NMR. When water is present with an alcohol, it will keep the alcohol hydrogen from coupling to the carbon it is attached to. So here, when we ran 1 propyl dry, we should receive a triplet and a doublet, but our machine is unable to resolve that. So when we ran it with D2O, which acts as water but does not interfere with the spectra, we get rid of our alcohol coupling and we will only see our triplet. We did the same thing with isopropanol. When we ran it dry, we expected to see our heptet of doublets, and when we ran it with D2O, where the alcohol doublet was taken out, we only see our heptet. All of these experiments and several others were typed up into worksheet format and compiled with the raw and interpreted data into binders for students to do similar experiments. Thank you for listening to my presentation over advanced NMR methods.